Good day, everyone. Our topic for today is about Module 5, Amino Acids and Proteins. I am Dr. Erwin R. Abrencilio. So for our learning um, outcomes for this module, 1. Describe the structure of amino acids and their classification. Second, explain how amino acids are linked together. Third, analyze the different levels of protein structure and describe how proteins are denatured. And fourth, distinguish the different functions of proteins. So let us now look into lesson 1, amino acids. So as we all know that the building blocks of proteins are the amino acid. So meaning for us to be able to understand better what proteins are, we have to understand also the functions and properties of the amino acids, since amino acids are the building blocks of proteins, or proteins are made up of amino acids. So as many as 300 amino acids occur in nature. Of these, only 20 known as standard amino acids are repeatedly found in the structures of proteins. So isolated from different forms of life, animal, plant, and microbial. So meaning, though we know that there are 20 amino acids in our body, or in the body of the human, in the body of an organism, 20 is just part of the 300 known amino acids. However, these 20 are repeatedly found in the structure of proteins from the different life forms. So meaning to say that these 20 amino acids are found consistently in the living organisms. So this is because of the universal nature of genetic code available for the incorporation of only 20 amino acids when the proteins are synthesized in the cells. So the 20 is patterned on the what we call as the universal nature of the genetic code. So as we know that the amino acids are likewise involved in that particular genetic code. So this is the generic or the general formula of the amino acids. So it has two functional groups. The first one is the amino group on the left side, composing of the nitrogen and two atoms of hydrogen. And then the carboxyl group, one atom of carbon, two atoms of oxygen, and one atom of hydrogen bonded on the upper oxygen. And then we also have the side chain. Um, the side chain is placed on the alpha carbon. So if you are going to recall your organic chem, alpha carbon is the first carbon next to the carboxyl group. So do not be confused, because most of the time when we speak of alpha, it is the first one. Yeah, it's the first one in that is next to the first carbon of the structure, or next to the carboxyl group. So let us now look into the classification of amino acid. So we have this classification. It is divided into two major classifications. So first is the structural classification of amino acid, wherein the one that will serve as the basis is the actual structures of the 20 amino acids, and then the nutritional classification of amino acids. So let us look into uh, for the structural classification of amino acid. So we have the following amino acids with aliphatic side chains, hydroxyl group containing amino acids. So hydroxyl group are the OH group. Then sulfur containing amino acid. So if the chemical structure of the amino acid has a sulfur to it, so it is a sulfur containing amino acid. And then acidic amino acids and their amides, and then basic amino acids. So there are acidic amino acids, there are also basic amino acids. And then aromatic amino acids, and then amino acids and heterocyclic amino acids. So these are those that I have mentioned a while ago. So these are the 20 amino acids as classified as non-polar aliphatic R groups on the first picture here on the upper left. And then polar unchanged group or polar uncharged group. So there's no charge at all. On the right side, upper, positively charged R group, and then negatively charged R group, and then non-polar aromatic R group. So the one that is being classified here is the R group, since the carboxyl group and then the amino group are consistently found in the structure. So that's why here in the non-polar aliphatic, so you'll notice that the alkane that are attached to the as a side chain attached to the alpha alpha carbon is that they are simple structure. So that's why this first classification sometimes is also called as the simple amino acid. However, aside that it is aliphatic, there's also an S here. So that means it is again still be reclassified into an amino acid that has sulfur to it. Okay, so we have glycine, alanine, valine, leucine, methionine, and isoleucine. So what I want you to memorize also is not only the name but also the symbol, the three-letter symbol. For example, glycine is GLY, alanine is ALA, valine is BAL, leucine is LEU, methionine is MET, and isoleucine as ILE, if I am not mistaken on the last one. Now, there's also one letter abbreviation. So it is part actually of your task that you have to submit at the end of the term, no? So upon the as stated in the slide. So you make sure to memorize the name of the amino acid, the structure of the amino acid, the what do you call that? The symbol, the three-letter symbol, and then the one-letter symbol. And then we have here the polar uncharged R group. So you'll notice that the R group, the one highlighted, has no charge at all. It is not negative, it is not positive. So it is uncharged R group. So they are polar. And then here is the positively charged R group. So you have there the NH3 positive, NH2 positive, NH positive. So you have here lysine, arginine, and histidine. And then negatively charged R group. So instead of positive, it is negative. So COO, CH2, COO, CH2, CH2, COO. So aspartate and glutamate. And then we also have aromatic. So when we speak of aromatic, it has benzene on the structure. So that's why here, no, benzene here, then another benzene here. So we have phenylalanine, tyrosine, and tryptophan. Okay, so this is the first classification. So lesson 2, another nutritional classification of amino acid. So amino acids are classified as essential or indispensable amino acid, and then non-essential or dispensable amino acid, and amino acid classification based on their metabolic fate. So let us now look into the first one, the 10 essential amino acid. So these are the 10 essential or indispensable amino acids. So I would like to highlight why they are considered to be as essential. Amino acids are known to be produced by the body of an organism. However, in the human body, there are 10 that cannot be produced by the body. It cannot be metabolized by our system. So that's why in order for us to be able to get it, we have to get it from our diet. So these are the 10 essential or indispensable amino acid: arginine, valine, histidine, isoleucine, leucine, lysine, methionine, phenylalanine, threonine, and tryptophan. On the other hand, these are the non-essential or the dispensable amino acid. It's okay to be dispensed. It's okay to be given to other. Since these are produced by the cell of our body. So we have glycine, alanine, serine, cysteine, aspartate, asparagine, glutamate, glutamine, tyrosine, and proline. So again, make sure to to determine the three-letter name as well as the one-letter name. So another is amino acid classification based on their metabolic fate. When we speak of glycogenic, we're talking about amino acid that will be turned that will become glucose at the end. If it is ketogenic amino acid, these are fats that are these are amino acids that are converted into fats. And then we also have amino acids that can do both. They can become glycogenic as well as ketogenic amino acid. All right, so let us now proceed to the peptide formation. As mentioned during the introduction or the glimpse or overview on the biomolecules, I mentioned that the amino acids are connected by a peptide bond. So the amino acids are held together in a protein by covalent peptide bonds or linkages. So formation of a peptide bond when the amino group of an amino acid combines with the carboxyl group of another amino acid, a bond is formed. Note that a dipeptide will have two amino group or amino acids and one peptide, not two bond. So peptides containing than 10 amino acids or decapeptide are referred to as polypeptides. So just like this one. So you have here on the left the amino acid 1 and then amino acid 2. The bond or the polypeptide or the peptide bond will happen between COO and NH3. Now, what happened here? So there is a removal of water. So COH and H3. And then after that, you'll be able to, when you remove water from the two, so one oxygen from COO and then two hydrogen in NH3, you'll be able to see when you remove it, it forms water as part of our metabolic processes. So CO will be connected to NH. And the connection between the two are known as peptide bond. In this case, this is dipeptide. Dipeptide because there is already a chain between two amino acid. So let us look into it. So this is the peptide bond. And then, for example, we have ala-cis-gly or alanine cysteine glycine. So you have here alanine, N, H is bonded to C. Ano, CO is bonded to N, to NH particularly. So this is the peptide bond. And then CO bonded to nitrogen here. This is again another bond, peptide bond. So how many, this is not just dipeptide, no, just like this one, dipeptide, but this could be tripeptide because there are three amino acids that are involved. So let us now look into the structure of protein. There are actually four structure of protein, so that depends upon on the functions of the protein itself. So not necessarily that all substances will go to the quaternary structure of proteins because some of them stayed at as a primary structure, some stayed as a secondary or tertiary, and so on. So secondary structure, the spatial arrangement of protein by twisting of the polypeptide chain. So that's secondary structure. And then the third tertiary structure, or the third structure of the protein, is the tertiary structure. So the three-dimensional structure of a functional protein. And then the last one, some of the proteins are composed of two or more polypeptide chains are referred to as subunits. The spatial arrangement of these subunits is known to be as quaternary structure. So to give you an idea, so this is how it looks like. So in the primary structure, it is still an amino acid. And then when it becomes more complex, it forms alpha and beta linkages, thus forming the secondary structure. Then after that, you have the tertiary structure, the three-dimensional structure of a protein. And then at the end of it is the last one. You'll see the difference, no? She can be able, that particular quaternary structure, no, is composed of several tertiary structure. So I think there are four. There's also secondary structure present. Okay. So let us now look into the classification of proteins for the last lesson for this module. So we have functional classification of proteins and protein classification based on the chemical nature and solubility. So this will be our point of discussion later on. So let's proceed to the functional classification of protein. So when you speak of functional classification, we are talking about the different functions of the amino acid composing the protein. So one is structural protein, no? So this is generic, no, not particularly one amino acid, no, but it's the entire protein itself. So proteins serve as structural proteins wherein keratin of hair, nails, collagen of bone. So those samples are for structural protein. Enzymes are catalytic proteins. So proteins also act as enzymes. Example are pepsin and hexokinase. And proteins also serve as a transport protein wherein they, for example, hemoglobin can transport oxygen all throughout the body. And then hormonal proteins like insulin and growth hormone, contractile protein just like actin and myosin in the muscles, storage proteins, ovalbumin and glutein are both storage protein. And then genetic proteins, for example, ay nucleoproteins. And then defense proteins, snake venoms and immunoglobulins. And then the last one is receptor proteins for hormones and viruses. So all these are the functional classification of proteins. So meaning to say that these are the functions, your functions as the functions of the protein itself. All right. So let us now look into the letter B classification, protein classification based on chemical nature and solubility. Under this are the simple, conjugated, and derived proteins. So there are a lot of simple proteins. So the first one is the globular proteins, spherical or oval in shape, soluble in water or other solvents, and digestible. So we have under that is albumin. Albumins are soluble in water and dilute salt solution and coagulated by heat. So examples of these are the serum albumin, the ovalbumin in egg, and lactalbumin in milk. So they can easily be coagulated or nagbubuo-buo sila, no? once there is an application of heat. And then we also have globulin soluble in neutral and dilute salt solution. We have betaline in egg yolk and serum globulin. And then gluteline soluble in dilute acids and alkalis and mostly found in plants. Example is the gluteline in wheat and oricinin in rice. And then prolamines soluble in 70% alcohol. Histones, strongly basic proteins, soluble in water and dilute acids, but insoluble in dilute ammonium hydroxide. For example, thymos histones. And then globins, these are generally considered along with histones. However, globins are not basic proteins and are not precipitated by ammonium hydroxide. We also have protamines. They are strongly basic and resemble histones, but small size and soluble in ammonium nitrate. So protamines are also found in association with nucleic acid. And for example, is the sperm protein. Okay, so these are the different, what do you call this? Yeah, it is protein classification based on chemical nature and solubility. And then we have also fibrous proteins. Fibrous proteins are connective tissue proteins lacking tryptophan. Tryptophan is another amino acid. So collagens on boiling with water or dilute acids yield gelatin which is soluble and digestible. Okay, so that means that we can be able to eat collagens. So elastins, these proteins are found in elastic tissues such as tendons and arteries. And then keratin in fibrous protein, these are the present in exoskeletal structures, such as that hair, nails, and horns in the animals, and the hair of humans that contains around 14% cysteine. For the conjugated proteins, okay, so the same thing. So for the derived proteins, we have the primary derived, secondary derived. Okay, so the primary derived are the denatured or coagulated or first hydrolyzed products of proteins. The other hand, the secondary are degraded. Degraded because they are made via the breakdown of peptide bonds. So under the derived protein, we have coagulated proteins. They are denatured proteins produced by agents such as heat, acid alkalis, and others. Cooked proteins, coagulated albumin, or egg white. So that's the coagulated proteins. And we also have proteins. These are the earliest product of protein hydrolysis by enzymes, dilute acids and alkalis, which are soluble in water. And then we also have metaproteins. These are the second stage products of protein hydrolysis obtained by treatment with slightly stronger acids and alkalis. And then this is the derived protein, secondary derived protein. We have complete, partially incomplete, and incomplete proteins. So for the complete proteins, these proteins have all the 10 essential amino acid. When we speak of partially, those proteins lack one or more essential amino acids. And then incomplete proteins, these proteins completely lack one or more essential amino acids. So hence they do not promote growth at all. For example, gelatin, it lacks tryptophan or TRP. So that would be all for today. So thank you so much, and do not forget to look into the details of this discussion in the module. And it is not included in the module the name, the symbol of the 20 amino acids. So you make sure to look into the name of the amino acid, the three-letter symbol as well as the one-letter symbol. Okay, so that would be all, and see you in our class.